Videos are. Okay, I'm going to show you the MCQs first. After which you can choose MCQs that you're studying. Was this already difficult? So, what would you say is because you don't understand whatever you learn in class? Okay, yes. Some parts are interesting, like the chloroplast, the plant cell question, right? So those questions, I understand. What about other questions like the electron transport chain? Sorry? Okay. I mean, we are all in the early phases of encountering questions that are for a particular topic, right? Now, I want to show you the same questions from your system. I can guarantee you that it's going to be easier because you've done it before, right? So, when you encounter new questions, I think a better way forward would be rather than I don't know how to do this. Let's reframe ourselves. We're going to find out how to do this. Rather than I don't know how to do this, we're going to find out how to do this. So when we say we don't know, we really box ourselves up. But when we say we're going to find out, the box is still open. You can ask your friends and search Google. First, I can guarantee you, if you want to type in the same questions in Google will give you quite a good answer. A video may pop up, a wiki may pop up, you all support your learning. Yeah. Okay, so we must adopt that mindset. Okay, don't box yourself in so early. Okay. So today, let's go through the MCQs first. Along the way, share some tips on how you can learn this chapter better. The MCQs start out with dog, boy, bat. Dog. Yeah, D. After which C. The last question, I felt like I used it on J1 before. So it's a photosynthesis question. I don't know primary school you got C before. Yeah. Okay. Somehow it's not like here. Okay, which question should we go through? First, second, third. Right. Hold on. Yeah, 8, 6 can give a plus now. Six. Okay, question 1. Which combination correctly describes light-dependent reactions of photosynthesis? Light-dependent refers to the electron transport chain. Okay. So where does that take place? Actually, is the thing random? The thing that sums you? No. Okay, then what would be better for you? If I say that the light-dependent reactions take place at the dielectric membrane area, is that more acceptable? Okay, so then the question is, what is random? Okay, no, your textbook. Your textbook, that's fine. What is random? Which page would you use? I really like to wear this shirt. I sweat like my armpit area like crazy. I don't know why. This shirt is so comfortable. It's not for my armpits. Where's random? Okay, so if you look on page 73 of the textbook, when we talk about random, we're talking about the sets of dielectrics. Okay, Yeah, so it's more encompassing. So when you find the electron transport chain, the location is at the diploid membrane. Diploid membrane is the membrane of each diploid. You will see a granum. Granum just means many stacks of them. All of them together, you find a granum. Yes. Oh, okay. The styloid membrane, the same as styloid lumen. Every time you see the word lumen, what do you think of? Hollow space. So give me an example of another lumen. Blood vessels. Blood vessels, if I were to slice a blood vessel and you talk about the tube, it's not even a thing, it's a space, right? So the space in the tube, there is the lumen. So lumen usually refers to the space. Yeah. So if I talk about diploid lumen, lumen is inside. The membrane is the over here, the membrane. So they are different things. Okay. So when we talk about the granum, in a sense, we are talking about both the space and the membrane together, which is okay. That's where the light dependent reactions take place. Okay. Where the light dependent reactions take place. Ah, I see. Okay, which combination describes the light dependent reaction? Oh, fair enough. Okay, I see where you're coming from. Yes. ATP.
Yes.
You
can see that it says ATP.
Yes.
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 And then it's ATP.
Yes. Yes.
And then that space is
Yes.
So I just use that and I use the answer. So I really don't
yes. Okay. Okay, I see where you're coming from now, Kitty. Because if you were to want to be very specific, ADP is synthesized in the stroma specifically. The light dependent reactions, all of the reactions, we will talk about the granum. Right? That one's still acceptable. But now let's talk about ATP. The talk about ATP, actually specifically is in the stroma. Yeah. Okay, so who put but there's no best answer therefore. The best answer will be D. If I choose any of the options with stroma, right, I also cannot because this one gets oxidized. Okay, so in this scenario, this is the best answer. But I see your point. I see your point there. Okay, that's a good point. Good question, no? Okay, question 2 is a question on the electron transport chain. So they ask what are the electrons and hydrogen ions used to produce. Maybe the first question we should try to address is what is reduced NADP and what is NADP? Okay, so in this case, when they say NADP, yeah, they mean NADP or plus. When they say reduce NADP, what do they mean? They reduce NADP, but they are referring to NADPH. That's what they mean. So what are the electrons and hydrogen ions used to produce? Is used to produce reduced NADP from NADP plus. That okay? That's the first one. We use electrons and hydrogen plus electrons plus hydrogen ions. That's how we produce NADP. We get NADP, reduce NADP from NADP. ATP from ADP. Is that okay? That's at the end of the electron transport chain. The end product of electron transport chain obviously. Okay, so that's question 2. Remember, some of the things you can do is to start. Anytime you encounter a question that you find would be useful to incorporate in your notes. When is your quiz? Is it? Next week, okay, so it's next week. You do have to start preparing your notes. Yeah? I think it's next week Friday, right? Yeah, so look at the schedule.
It just looks like it's next week.
Okay, it's next Tuesday because Friday is Good Friday. So I will not see you on Friday. Okay, it's next Tuesday. You can start preparing for your notes already. Okay, so your notes is a combination of respiration and photosynthesis. Do you need 2 sides or 1 side? I was thinking 1 side. It's actually possible. Yeah. Okay. Let's try 1 side. If you try to squeeze in 1 side, right, the chapter will become clearer to you. Okay. Photosynthesis, cellular respiration study together. Okay. Next question, question 3. Weed killer. Sounds like some WWE fighter in the ring. Okay, the weed killer blocks the flow of electrons, the light-dependent reaction of photosynthesis. Why does this kill the plant? It kills the plant because the products that are produced from light-dependent reaction cannot be used for the Calvin cycle. The Calvin cycle is used to produce glucose.
What is C?
Okay, so I see not correct. Okay, so why can't we accept C? Photoactivation of chlorophyll cannot occur. So if this is electron transport chain, I have those photosystems with chlorophyll inside, and then there's light striking the chlorophyll. Can the electrons be activated, excited? They can. Nothing's stopping them from being excited. However, one thing is now an issue. They may be excited, but they cannot be passed on. Okay? This cannot happen anymore. You cannot pass it on, but can still be excited. Nothing stopping them from being excited. ATP and reduced NADP are not produced. Actually, that's a problem. If this whole thing doesn't function, no more NADPH, no more ATP produced. And these 2 things are so important for the Calvin cycle. Okay, but cannot function. Next question. Which is an electron donor for the light-dependent reactions in photosynthesis? It is water. It comes from water. In the case of cellular respiration, oxygen accepts electrons to become water. This one, water, is the one that is up. Okay, so maybe I make it clear to you for your quizzes, right? Every time we have a quiz, I will highlight certain names. If your name is highlighted, it's suggested that maybe you want to join Knowledge Cafe so you can top up and help each other out. Yeah. I told those that attended yesterday is that our mission this year is very simple. We be consistent. Yeah, that's all we are doing here. How often do you make your notes on the phone? Right? Last time you ask yourself, how often do you make notes on the phone? Any subject you do that for? But this year, if you look at biology, you are making notes on the phone. Granted, the notes may not be the best, but it forces you to think a little bit harder about what you've learned in the moment. Yeah, rather than do that 3 days before the exam. Right? My handphone usually non-stop, but there are text everybody. Yeah. Question 5. Question 5 is a question asking you where water and carbon dioxide is. That's all they're asking. So question 5. Question 5 If I rephrase the question, I ask you, where is water used in photosynthesis? Where is CO2 used in photosynthesis? The thing about this experiment is interesting. What they did is they used water that is made up of oxygen that is heavier than typical oxygen. A typical oxygen is 16 mole, right? So 18 mole is heavier oxygen, this water is heavier. Therefore, it's also radioactive, it can be detected using instruments. CO2, this particular CO2 is made up of a C that is C13, it's heavier than usual, it's C12. And the oxygen is also heavier, C417. So we are using CO2 that is heavier and water that is heavier to conduct this experiment. We're going to find out where water and carbon dioxide is used in the plant cell. Where is water used? Water is used as an electron donor for photolysis to occur. If you want to find out where that takes place, look into your textbook on page 78 of the textbook. You can find the equation where H2O is converted to O2. You could highlight that so that you can find out the location. The location is in the thylakoid space. Yeah. Where is CO2 used? It is used in the Calvin cycle. So the location would be in the soma, where Calvin cycle takes place. For water, it is used in the thylakoid membrane. Thylakoid space. What's interesting about all the MCQs so far? They've been testing you 2 things: location and product. Say again, perhaps one form of notes you can make for this chapter is when you have a table of location and product. Let me give you a sample of the set of notes you can make. Perhaps you could try something like that. You could do something like lipolysis. Then you put location. Then you put products. Then you do a table. That's one way I will make notes to know the location of products. Then if I do MCQ or ask you any structured questions, you can immediately test it. Yeah, so that's one way to compress everything into a table. Last question, question 6. Perhaps you can talk to your friends. I have no idea what time. I think it is 12 o'clock. So I've been panicking inside. Can we pause a lot? Take any questions if you have. Let's go to structured questions. Okay, question 1A. We are asking the let's take the importance of photosynthetic pigment sustaining light. You know what photosynthetic pigment is? No. Okay. Let me give you an example of photosynthetic pigment. An example of such a pigment is chlorophyll. Okay. So an example of such a pigment is chlorophyll. So this table shows all the types of photosynthetic pigments they can find. Some of them on Earth. Yeah. I say it as though you can find others on other planets. These are the photosynthetic pigments you can find. Believe it or not, some bacteria have chlorophyll, just their version of it. What's unique is that their version of chlorophyll is not green, it's purple, like your shirts. So these bacteria look purple color. Yeah. Carotenoids is another form of chlorophyll. If I'm not wrong, they are the ones that look reddish. They result in sometimes a reddish tinge in leaves. Okay, it's just different kinds of chlorophyll. Okay. Yeah. So different amounts of different chlorophylls result in different colors you see in the plants. So, what is the importance of chlorophyll? I will rephrase the question. What would you say? What is the importance of chlorophyll? Not the harvested. The question is asking state the importance of it. Okay, it's important for photosynthesis, but more specifically, what is its role? Okay, I learned a few things. I hear make up electron transport chain. Absorb light energy so that it can be used for electron transport chain. Anything else? For photolysis. Okay. Photolysis actually is done by the enzyme. Yeah. Not by the light per se, but it's done by the enzyme. There's an enzyme that helps to carry out photolysis. That's a great question. Not sure. Yeah. I suspect it's because, yes, it needs light. But then again, you see, they're asking about photosynthetic pigments, they're not asking for the importance of light. Yeah, I'm asking for the importance of the chlorophyll, not the light. Okay. So, where were we? You were saying absorption of light used for electron transport chain. Anything else that we could use in this to supplement this answer?
Can we say what?
That's interesting. I would say that won't help to answer the question. What do you say? Yes. Okay. Okay. Yes. Yeah. Okay. Then let's draw the final link. We say that there's light absorption. Okay. That's one thing. But how do we link this to sustaining light? Cannot make food, right? So actually there are two parts. Number one, you cannot get light. And this, therefore, my electron transport chain cannot function. And downstream, I cannot produce what I need, which is glucose, which is food, which sustains life. For biology, just do your best to link back the questions then, whatever they ask you at the beginning. So, absorb light energy to make food. Starch. Did anyone talk about oxygen produced as a byproduct? That's important for life also. Yes. It is. But the ATP isn't used directly by the plant for energy. It is instead channeled to produce glucose. They do. So the glucose that they produce, they will then channel the glucose to their own mitochondria, carry out cellular respiration. Alright, good question. So I think last week or week before, Sunday tried to address that question. So we have a very weird issue here. The chloroplasts during the light dependent stage, they can produce ATP. This ATP will later be channeled to produce glucose. Then this glucose will be given to the mitochondria of the plant cell to then produce ATP. It's very weird. If the plant can produce a little bit of ATP at the start, why must it use it to produce glucose, then pass to mitochondria to produce ATP? Okay, so actually many reasons. One is this glucose is a good form of storage. And to store it until I want to use it to get more ATP. And then Bansi brought up one point interesting. At night, no? At night there's no light. At night the plant actually needs food. That's when it can use all the glucose is stored to get ATP. Okay? Yeah. So that's quite interesting reason. All of this is also on Google Classroom. Yeah. If you ever lose your worksheet now, you can find digital versions. Okay, so the question asks, suggest and explain where you can find light harvesting complex in the bacteria. What a weird question to ask. This question really pushes you to challenge you a little bit. Suggest it is not out of your realm of knowledge, but it's challenging. If you can use anything you can think of. Where do you think you will find the electron transport chain or the proteins? Where do you think you'll find it when I look at bacteria? Bacteria sometimes got flagella. Let me give you a clue. Okay, where do we typically find the membrane proteins? Okay, on membranes, right? Bacteria, let's think back up. Bacteria are like regular eukaryotic cells. They don't have membrane-bound organelles. So all they have, right, is a bunch of floating DNA inside. But they have no mitochondria. Yeah. So bacteria, they do not have membrane-bound organelles. They don't have a mitochondria inside. They don't have any other membrane-bound organelles inside for you to put the membrane proteins or electron transport chain. So where would you think you find the electron transport chain? Yeah, right. It's only one place left. Actually, the light harvesting complex or the membrane electron transport chain they're all found on the surface. So you know actually scientists speculate. We theorize that the mitochondria we have used to be free roaming cells. So imagine now I box up the cell and I keep one of these bacteria within me, within my cells to create the ATP for me. That's the theory. The running theory is that mitochondria chloroplasts use the free roaming cells that can produce food, produce energy. Yeah, bigger cell came along to adopt them, and then they partners ever since. That's the theory. They use these cells. Why don't we have chlorophyll? It's a great question. I don't know why you have chlorophyll. Oh, but one student from another class speculated that perhaps it's not efficient. Okay, define efficiency. Enough what? Okay, chains. You think what do you think about chains theory? But photosynthesis cannot produce enough glucose per unit time.
We don't need glucose.
But you think yourself as an animal, that needs to do a lot of stuff, right? And then compare to a plant that doesn't move.
But then we have so many cells.
Exactly. We have, okay, but how much of your cells are actually exposed to the sun? Let's take all those notes up, right? And then we look at a plant. The plant doesn't move. So actually the glucose it produces, right, is enough for it. It's a sedentary lifestyle, right? And you look at us as animals, actually you don't just eat one leaf. We eat many plants, right? to be able to get enough glucose, enough energy. So perhaps this is one reason. What Jean said that we don't use this process because it's just not enough for us. Maybe. Okay, so where can we find it? A light harvesting complex of the cell membrane. Because the light membrane. Not that I recall, I do know of bacteria that have cell wall, but more than one cell wall. I'm not exactly sure if there's more than one cell wall. Google and Google it. Yes. Bacteria actually have cell wall. Bacteria just come with cell wall. They ask you to identify one pigment that can be found in purple bacteria. To answer this question, you have to look at this. This graph shows you how much light is absorbed by the chlorophyll. Will you tell me which wavelength of light is this plant absorbing the most of? So 850 seems to be the most of the region of wavelength of light is absorbing the most of. So let's look at the table. At the table, which chlorophyll pigment allows the bacteria to absorb light at that wavelength of light?
Chlorophyll A.
Which one?
The bacterial chlorophyll A.
Okay, A seems to fall in the nice range. Okay, so that is the answer. A. Let's suggest and explain an advantage of synthesizing photosynthetic pigments of chlorophyll that's different from the plant chlorophyll. So, okay, this question is difficult. Every time you see a questions you suggest, right? Prepare yourself because this question requires you to be creative and imaginative within the realm of biology. Okay, so let's listen to your answers. Suggest an advantage of using different kind of chlorophyll than the plant chlorophyll. Why would bacteria use a different kind of chlorophyll rather than the green chlorophyll that plants use? Why don't they just use the green chlorophyll? Tanya?
I know that
Okay, so Dania is explaining an advantage of using different pigments is to tap on other wavelengths of light. You see tap on other or tap on more wavelengths? Different wavelengths. What's so good about tapping on different? Ida is, yes, Ida. Okay. Okay, so Ida's suggesting that perhaps in the deep waters where the light that plants normally use, perhaps it's not reaching it. Therefore, perhaps having different chlorophyll will help us to harvest that light. That sounds reasonable. Any other suggestions? So let's have a look. I don't know what I see wavelengths or absorbed by chlorophyll are not able to penetrate the waters. And that's what Ida said. We use competition for same wavelengths of light. Let's say the light that plants use can reach the bottom of the ocean. I guess we don't compete now. Plants, you can use your fair share of light. I use my own. Then we use different wavelengths of the rainbow. Yeah, that's true. So there you would reduce competition only if they can exist in the same place. If they don't, if at the deep water, deep one, right, only bacteria don't touch, actually some condition. Then the second point will be more relevant. But if I would bring the bacteria to the surface, where plants are, then there's no competition also. Long day trade-off. Friday a long day. Always what? Hot.
Yeah, it's always like really sunny.
What's the role of PQR? The role is they serve as electron carriers. Anyone got anything else? Aid in flow of electrons. Okay, so how we describe it as? Aid in the flow of electrons. I think I would be okay with that if you leverage a little bit more. Aid of flow electrons through the electron transport. Then I know the context where it's happening. Yes. Earlier you had what a suggestion. Yeah, I think that's okay. Yeah. Go.
I say they oxidize NADH
Oh, they oxidize NADH.
and FADH.
They are. Oh, maybe firstly, NADH and NADH2 is for cellular respiration. So this is photosynthesis. So let's perhaps talk about NADP. Okay. So you're saying what if we say they used to oxidize NADP, right? Actually, we cannot oxidize NADP. Oxidize means to take away electrons and hydrogen. So maybe NADPH. If I want to oxidize NADPH, that wouldn't be the role of the electron transport chain. All they do is to, after this has been, actually this is the final electron acceptor of the photosynthesis of light dependent stage. This is at the end of the electron transport chain. So all the proteins, the electrons pass to this, then it becomes NADPH. Yeah. So to say that the function of this is to oxidize this, which also cannot. Once you're passing electrons to this, you're reducing it. Yeah. So actually, and also, this one doesn't directly or reduce this either. Their role is probably to pass electrons. Good try. So explain the impact of photosynthesis if Q is inactivated. What happens if I knock out Q? Okay, can you prepare yourself for electron transport chain for this chapter? Mentally prepare yourself for a question that will ask you, what happens if I knock out one of the proteins? Yeah, just prepare yourself for that. Okay, every RE we encounter something like that. Your quiz, where it happened?
Yes.
Maybe. Okay. So explain the impact. The usual things. Electrons cannot be transported. But in photosynthesis, it's a bit different. It cannot be transported from photosystem to photosystem to replace the electrons that were excited. Okay, that's one thing. NADPH cannot be formed. So what it goes when it U2. Electron transport chain ceases to function, like cellular respiration. No proton gradient. No proton gradient. ATP cannot be synthesized. And if ATP is not synthesized, in this case, Calvin cycle cannot function. You cannot get glucose. Yeah, just 4 points. Only 1 point. So for biology, you see a 4-mark question, aim for 4 points. If you got time, you put in 1 more point. You see a 4-mark question, put 2 points, and be very sure that you will not get the other 2 marks. Right? so that's how I usually think when I see the marks allocated.
We need to talk about the photon radius if they talk about
But there's still a photon gradient, right?
Kelvin cycles.
Yes.
Can I just talk about any? Can we not talk about?
It's crucial. It's crucial. Every time a protein has someone at ETC, these are the usual suspects that you yes. Oh, okay. Oh, but you mentioned ADP. Okay. No, I also not so okay because the products of the electron transfer chain are these two things. Fair enough. Well, I wouldn't avoid you the mark. That's not so. So it's fine. At this point, not so much about getting the marks. At this point, we find a way to place our answers. But these aren't model answers. Actually, if you look across the RAs, the way we place points 3 and 4 differ a little bit here and there. But the idea is the same. Then the next question is the question that maybe I'll find. Okay, for those that did not attempt some of the questions, while waiting for a fix, maybe now's the time to try. If you're not attempted, now's a good time to try. No, you all weren't born to fail. When you were born a baby, all of us are born babies. When you first born, the fact that you can walk today, right? It's a testament to how you will never quite give up when you were young. Which baby after standing up the first time, then they fall, right? Then I'm gonna sit here, I'm not gonna walk anymore. That's it. Don't. The fact that you are walking today is a testament of that. Yeah. But could be a bit. Okay, so what's the code? You weren't born. You are just a okay, let's go to this question. This question. But it's not your textbook. So let's try. Okay. How at least you mentioned chloroplast. Okay. That's one thing you could try to do, right? There's chloroplast. First, chloroplast, one of its roles is photosynthesis. So you could mention chloroplast for absorption of light. The other 2 points are not too far-fetched. Okay. Let's look at first point. Cell wall, cell membrane. Actually, they are permeable to allow nutrients to go in and out. Products go out, nutrients, raw materials go in. Large central vacuole, if you recall away from H2, large central vacuole, the purpose is to store sugars. Store not so much sugars as such, 10 sugars. Actually, store sugars, not sugars, not starch. Yes.
has water, right?
Yeah.
Sometimes it is big, it is the center to supply water all the time.
Yeah.
And that is the plan.
Oh,
Plus it's so big.
store water for chloroplast.
The vacuum is big and it is in the center to supply water for the plants.
I think that's small. If you want to be more encompassing also, you could say that the large central vacuum therefore can perhaps store maybe water and carbon dioxide. Carbon dioxide don't store it. But I think that's reasonable.
Is that why, like, that's why when you pour a lot of water,
Yeah.
I mean, the sound
It gets touched, right?
because the vacuum is
Yeah. Okay, but I do have to say though that the function is not so much to store water for that. It stores water to keep the plant fertile. Yeah, that is the main function. So I wouldn't. It's not. It doesn't really store it per se for photosynthesis. Yeah. That is okay. Yeah, I think that is reasonable. Yeah. But the cell wall is possible. Yeah. You can have a thin or thick cell wall. Yeah. But cell membrane, you're right. Actually, it's just bilayer, not thin or thick. Okay. So the thin, I think, is referring to cell wall, not cell membrane. Thin cell membrane. Thin cell wall. Cell wall can be thick or thin. Just how much cellulose we access. Last question. Last question is asking you, what is the role of the thylakoid membrane in photosynthesis? Okay, this question seems, when you first look at it, it seems quite difficult. But actually, with showing whatever you know about thylakoid membrane, you will help you. Okay, so thylakoid membrane, what do you find that? Electron transport chain. They threw that in one bucket. So what else may you find in thylakoid membrane? They produce ATP, right? Okay, so what will you find that? ATP synthase, 2 marks. Okay, well, so the idea here is to talk about thylakoid membrane. You find out the proteins, all the proteins involved in the transport chain. Then you can set up the proton gradient, the usual things. So let's have a look at. Okay, the first point you may not have is that it's a large surface area. You have a lot of the membranes used to make many thylakoids. Contains a lot of photosystems. Photosystems are what contain chlorophyll. We get to check harvest light. You mention all the proteins of the electron transport chain. You mention proton gradient. You mention ATP synthase. So the usual suspects. This is a hot favorite question to see if you understand the topic. Okay, hot favorite. Be structured, execute, essay, hot favorite. Second and third point, what's the difference? Okay. Electron carrier and photosystems, they are all part of the electron transport chain, but their functions is different. Yeah. The electron photosystems are these fellas, you know. And then they have chlorophyll pigments. Their role is to harvest light. These are photosystem here, photosystem 1 or 2. The proteins, their role is just to accept the electrons and pass that on. But these fellas absorb light, to excite electrons. Okay, so their functions are different. Therefore, we put them as separate points. But we combine it in one sentence. Yeah. Okay. If you didn't mention emit high energy electrons, another way I could phrase it, or you could phrase it, is that we absorb light to excite electrons. It's another way of phrasing it. Yeah. But you do have to explain the function of it. That was why I have so many systems. Okay, last one. Describe how carbon dioxide is fixed. Just mention and you use this enzyme. The enzyme is Rubisco. I don't even know the long name. Okay. You mentioned that you need this enzyme. Okay, that's all there is. Then all the best in your note making, and we will refine your notes.